Well, I want to thank everybody for coming and thank Paulo for inviting me. As he said, we've been good friends for a long time and visiting here and him coming to Montana. And so today I'd like to talk about what does effective population size tell us about the loss of allelic variation. And this is my co-author, Nils Ryman, and Nils and I have been friends and collaborators now for 49 years, which is amazing to say for me. And so this is a paper that we just submitted that is part of the special edition of Evolutionary Applications. So this is in honor of Mike Bruford, who died a couple of years ago of cancer. And I only met Mike twice, and once was here at a meeting 8 years ago, and the other was in Namibia in 2016. So in honor of Mike, they organized a special edition, and we submitted a paper talking about allelic variation and what does an E, effective population size, tell us about allelic variation. So this is all of my talks. I guess I always get up in a soapbox and talk about the importance of, well, theory in population genetics and in conservation genetics. So this is the book that Paulo mentioned that was published a couple of years ago now. And my whole career has been spent really trying to apply population genetics to issues in conservation. And for me, this has been really rewarding because, in my view, some of the most interesting questions, basic questions, are also the most important questions for conservation. And I like to think of empirical population genetics as being a stool with three legs. So you have to collect data, you have to do some kind of analysis. But to me, the really, not the most important, but the thing which I think has been sort of left behind is theory. So people now, much more of the training is in analyzing data and collecting data. But I think it's really important to understand the theory before you can really apply these data. And so this book by Svante Pääbo, which I think really says it well. So obviously he was a leader in going out and getting genetic data from fossils 40, 50,000 years ago. But to getting the data is one thing, but understanding the data is another. And this is from the book where he talks about the importance of collaborating with population geneticists like Monty Slatkin, who really can take those data and interpret and tell us what those data really mean. And a couple of reviews of our book, which I think is interesting because obviously the reviewers got it. We spent a lot of time in the book. It's really a population genetics book with applications to conservation. So the Linda Neves review talks about the solid background of theories and concepts and how this is really strength of the book. And then the other review, which is interesting, which says that this application to theory and mathematics may be challenging for readers without an advanced understanding of mathematics and genetics. But I really think if you don't have that level of understanding, you shouldn't be doing population genetics because it requires that to really understand what you're doing. And this book is still probably the best book in theoretical population genetics. It was published in 1970. Jim Crow was a really wonderful person and a wonderful population geneticist. And it's still available for $55. It's been reprinted. And I always say this at my talks, but for this particular paper, we really relied on this book. A lot of the formulas I'm going to present to you came originally from this book in 1970. And Joe Felsenstein says things a little bit more colorfully. We had the same situation in population genomics. People have vast amounts of data and do completely half-assed things with it because they don't know any better. And I wish there was some way of persuading people that we need to train students in development and properties of the methods, and that means population genetics. Oh, one more thing, if I can go backwards. So this is Joe's book, which he originally started when he taught population genetics, and that's the last, he puts that online. It's really, I think, the best up-to-date book on population genetics theory, and you can download it for free if you just Google Joe Felsenstein. So this is a problem or an issue that I've been working on since 1986. So I went to a conservation genetics meeting in sometime the mid-1980s, and people kept talking about heterozygosity, and they were making a lot of recommendations, which I thought were a problem because they didn't deal with what those recommendations said about the amount of allelic variation in a population. And so when you measure genetic variation in a population, there's two things that we're measuring. One is heterozygosity, and the other is a number of alleles. And so that was the origin of this paper. And this is just an example showing the basic fundamental concept is that, so this is a bottleneck of 10 individuals. And I started with a population with either 2, 5, or 10 alleles that were equally frequent, and then simulated what happened over time. And the dotted line is heterozygosity, which we can predict. We lose 1/2n of the heterozygosity each generation. And the heterozygosity is really nice because it doesn't matter how many alleles are present initially. Heterozygosity will always decrease by 1/2n per generation. But you can see, depending on the number of alleles and the frequencies, we get a different result. So if we have many alleles at a locus, we lose much more genetic variation than we do compared to heterozygosity. So one of the basic points of my talk is that heterozygosity is much less sensitive to bottlenecks than is allelic diversity. And Paulo mentioned that Gordon Lukart was my student, and Gordon was a grad student in my lab right after I wrote this paper. And he took this idea of the effect of bottlenecks on heterozygosity in alleles and ran with it and did a number of papers. And he and Jean-Marie Cornouillet wrote the paper in 1998, which used this observation that allelic diversity is much more sensitive to bottlenecks than is heterozygosity, to develop a test when you have empirical data to see if you can detect a bottleneck in a population. So these are some simulated data that they did, taking microsatellite data from brown bears in Alaska, and same kind of plot, amount of variation remaining over time, over 5 generations, and this was a bottleneck of 2 individuals. So again, allelic diversity is much more sensitive in general to bottlenecks than heterozygosity. And this is one application of that test that they came up with, which is still being used in the literature. So this is work done by another student of mine, Christina Ramstead. She worked on sockeye salmon in southwest Alaska. So Lake Iliamna is a huge lake which has millions of sockeye salmon spawning every year. Lake Clark is upstream from Lake Iliamna, and it only became deglaciated a few thousand years ago. So it was a bottleneck population that came originally from sockeye salmon and Lake Iliamna. So Christina had collected both at that point, allozyme data, microsatellite data, and mitochondrial DNA data to look to see if she could see a signature of that bottleneck in these populations. And this is the basis of the test that Gordon and Cornouillet developed. So if there's theory that allows you, if you look at the number of alleles at a locus, when you collect data from the natural population, you can estimate what is the expected amount of heterozygosity we expect in that population at equilibrium. And this is called the mean heterozygosity observed, and this is the mean heterozygosity expected based upon the number of alleles. So Lake Iliamna are these open triangles and circles, and that's the source population. And the closed ones are Lake Clark, which were the ones that were bottlenecked and founded about a few thousand years ago. So you can see both for allozymes and microsatellites, the points for the Lake Clark, the founded populations, the heterozygosity is in general greater than that we expected based on the number of alleles. So this was the bottleneck test that they developed. If we look at a population, collect data, And see, we see more heterozygosity than we expect, given the number of alleles. That's a signature of a bottleneck. So this talk is focused around effective population size, and it's probably familiar to everybody, but I thought there are many different definitions of effective population size. So I'm talking about the inbreeding effective population size, which is based on the heterozygosity will decline at a rate of 1/2n per generation, and inbreeding will increase at the same rate. So heterozygosity will go down 1/2n, and homozygosity will go up. And as Sewell Wright said, whatever we have to put in this equation to make it work. So if we go out and look at a real population and look at the decline in heterozygosity, by definition, NE is whatever we have to use to make that amount of heterozygosity decline. And this is commonly applied to conservation, and the 5,500 rule is still used. This was originally presented by Ian Franklin in 1980, and Ian said that if we have an NE of 50 or less, that is too small, and if we have an NE of less than 50, inbreeding depression is probably going to play a role in the probability of that population persisting for the short term. In the long run, an NE of greater than 500 is necessary to make sure we have enough variation remaining. So variation is based upon the number of new alleles or new variation put in by mutation and the amount removed by small effective population size. So Ian Franklin made the comment that NE in the long run should be at least 500. And this is commonly used in lots of applications in conservation. And the point of my talk is that this doesn't tell us everything about genetics. This talks about heterozygosity. It tells us nothing about allelic variation. And the question that I want to address today is, what does this tell us? What does NE tell us about loss of allelic variation? And so there's these two things we're going to look at, heterozygosity. So at equilibrium, the heterozygosity is a function of NE, the effective population size I just talked about, and which is removing variation, and mutation, mu, which is introducing variation. And again, heterozygosity is really nice and simple. This is the expected amount of heterozygosity in a population as a function of NE and mu. However, the number of alleles is much more complicated. So we have NE, which is the effective size. We have NC, which is the census size, the number of adults in the population. And at equilibrium, the number of alleles, unlike heterozygosity, is a function both of NE and census size. So this is much more complicated, and it takes into account both the number of individuals and the NE. And that's one of the take-home messages from my talk, is that NE doesn't tell us everything. And if we want to think about the number of alleles in a population, census size is also important. And this just a graph showing that. So the x-axis is heterozygosity. So this is with a mutation rate of 10 to the minus 5. And this is the amount on the x-axis, and the y-axis is the number of alleles that are present. And the big thing to see here is that the number of alleles is both a function of NC and NE. And the larger NC is, the more alleles we expect with the same effective population size. And it's really curious to me, this is, Motu Kimura published this in 1964, but it really has sort of been ignored since then, and people don't always appreciate the fact that the number of alleles is a function of both of these. And this gets back to what I talked about before, the effects of bottlenecks. So we lose, by definition, we lose 1/2n the heterozygosity per generation, which is nice and simple. It doesn't matter how many alleles or their frequencies. But heterozygosity is really insensitive to small bottlenecks. So for example, if we have an NE of 2, we only lose 25% of the heterozygosity. So that means that based on this theory and a lot of assumptions, if we take Adam and Eve from the humans and start a new human population, heterozygosity would only go down 25%. But obviously, we're going to lose a lot of alleles because there's two individuals can only carry at the most 4 alleles. So heterozygosity sort of diminishes the effects of bottlenecks in a population. And as you probably got the idea now, allelic variation is much more complicated. So if we have A alleles in a population now, and we go through a bottleneck, the number of alleles left after the bottleneck is a function, the number of alleles and the frequencies, which is P of those alleles. So allelic diversity is much more complicated than is heterozygosity. And this is actually the same plot I showed before. So this is the paper now. I used EasyPop, and we did the same kind of simulation. So dotted line is loss of heterozygosity. And the more alleles that are present at a locus, the greater the loss of allelic variation relative to heterozygosity. And this is a function of rare alleles. If we go through a bottleneck, it's very likely that rare alleles are going to be lost. So for example, if we have an allele at a frequency of 1%, and we go through a bottleneck of 5, 10, 15, 20, or 25, even with 25 individuals, we're most likely going to lose an allele which is at a frequency of 0.1%. And this is another bottleneck test that has been used. So this is again Christina Ramstad, but this time working on kiwi and not sockeye salmon. So kiwi is well, these are little spotted kiwis. There are about 5 or 6 different species of kiwi. And all of the little spotted kiwi in the world removed the Kapiti Island about 50 years ago. And a number of subpopulations have been founded from Kapiti on these other islands. They're all in islands off of New Zealand. And so if we look at the allele frequency spectrum, so we can see in Kapiti, which is the source population, we see, so this is allele frequency going from 0 to 1. So on Kapiti, we have a number of alleles. Actually, the most common alleles are at 2% or less. But if we look at the birds on Long Island, these allele classes are empty now because they were not found in the founders. So since there's only 3 founders, the minimal allele frequency is 1/6, because there were 6 genes in those 3 founders. So the minimal allele frequency is down here at this point. So the rare alleles are lost if we go through a bottleneck. So if you have any questions, stop me either in human or remotely. So this is the basic idea is this differential effect of small populations on these 2 measures of variation. So why do we care? What difference would this make in terms of conservation? So one thing we're interested in is a population being able to adapt to new diseases, to environmental change, to other kinds of changes. And heterozygosity is a very good measure of, well, it's related to additive heritability, which is a measure of response to selection. So the higher the heterozygosity, the more likely populations are going to be able to respond to selection in the short term. So heterozygosity is a good measure of a population we evolve in the immediate future. However, allelic variation is not very important for short term. So rare alleles are not going to contribute much to the short term adaptation because they're so rare, they're not going to have much, they're not going to be, their increase in frequency is going to take a long time. But in the long term, having allelic variation is really going to be crucial. And this is another take-home message that heterozygosity, a good measure of what's going on now, but if we're worried about the future, we have to also worry about the number of alleles. And when people talk about using effective population size, one of the things that we do is we correct for different. So in an ideal population, we have equal numbers of males and females. However, if we have unequal numbers of males and females, we can estimate the loss of heterozygosity, which is based upon NE, by using this familiar formula. But this plot shows that we get a very different result of a bottleneck based using that equation. So here we have two pops. So here's again loss of heterozygosity, the dotted line. If we have a census size of 2 males and 2 females, this is the result of simulation, looking at different numbers of alleles. And C is a population with 99 females and 1 male. So if we use that NE formula, both of these populations have an NE of about 4, but we see a much greater loss of variation of allelic diversity in the population with a larger census size. So well, to me, it's helpful to have examples. So this is an example. So let's think we have a population of trees, a forest of trees, and we cut down all of the trees except one. Then we found a population. Obviously, this is an extreme example, but I think it shows the point. Let's say that we start a new population just from the seeds that are in that one tree. What is going to happen to heterozygosity, and what's going to happen to allelic variation? So what's going to happen to heterozygosity in this population founded from the seeds on one tree? What is the effective size of this population, which tells us what's going to happen to heterozygosity? Anybody have any guesses? Can you speak louder, please? And then I'll try to repeat what you said. Sorry. Maybe you need the box.
Okay, can you
Yes.
everybody hear me now? So I was just saying, after the trees have been cut off, the only tree that stands there will have the heterozygosity of the population, because
Okay.
obviously they were all the same population. But obviously that tree will not have all the alleles of the population.
But
just
we're going to start the next generation from the seeds on that tree.
Yes, so
what's going to happen to heterozygosity?
it will be the same.
Of the next generation with one mother? NE is going to be 4. So using that formula, if you have one mother and 1,000 fathers, the NE is going to be 4. We lose 1/2n, so we're going to lose 12.5% of the heterozygosity because we only have one mother. But now, what's going to happen to the number of alleles?
How many you were saying the seedlings? How many?
Well, so this is a pine tree, has 1,000 seeds, which is a random sample of all the trees in the population which pollinated those seeds. So how many alleles do you think are going to be lost? I would say probably none, or very few, because those seeds, they assume there's random mating in this large population, are going to be pollinated by lots of different males. So just like the theory here, so heterozygosity is going to decline very quickly. This is going to have an NE of 4, so this is what's going to happen to heterozygosity. This is when we have, I should have done this the other way around. This is, say, one female and 99 males. So we're probably not going to lose any alleles. So this is a case where we're going to lose more heterozygosity than alleles, again, because of the bottleneck effect and the fact that there are so many males contributing to this population. So another way that we think about effective size is over time. So if we have population any changing from generation to generation, this formula will give us a harmonic mean, which will allow us to predict the loss in heterozygosity. But what's going to happen to allelic diversity? Allelic diversity is going to depend upon whether we have one extreme bottleneck where we're going to lose almost all the alleles, or we have a larger bottleneck over a long period of time. And if we have an extreme bottleneck, we're going to lose much more allelic diversity with the same NE than if we have a bottleneck where the NE is small but constant over time. So again, looking at changes over time, NE that we use to estimate heterozygosity is not going to do a very good job of describing the loss of allelic variation. So the basic point is that NE doesn't do a good job in describing loss of alleles. The question is, does it matter? And there are two cases that I'd like to argue that when it's going to be most important. One is if we have a locus with many alleles. So as we saw, the more alleles that are present, the more allelic diversity you're going to lose. And the second is, based upon that slide that showed, if we have the same NE, but a larger NC, we're going to have more alleles. So this is the second case where effective size is going to do a poor job of telling us what's going to happen to allelic diversity. Whoops, I went the wrong way again. So there's three cases. So talking about alleles with many loci with many alleles, MHC in animals, R genes in plants, and S alleles in plants are three examples of loci where we have a lot of alleles present that NE is going to do a very poor job of predicting the effects of a bottleneck. So this is one example of penguins that was published about 12 years ago. So looking at one MHC locus, they looked at about 50 males and 50 females. And in the 50 males, they found 28 alleles. In the 50 females, they found 25 alleles. In the 100 birds total, there were 45 alleles present. So MHC is under strong selection for allelic diversity because of disease resistance. And so this is a locus in lots of species which has been found to have many alleles. And looking at this particular study, they looked at the fitness of females that were either homozygous or heterozygous. And they found out the females that were heterozygous had much higher fitness, both in the number of eggs hatched, the dark lines, and the number of chicks fledged. So MHC is a locus that NE is going to do a particularly poor job of describing loss of variation, and it's one that has great importance for adaptation. Another one that I mentioned was R alleles in plants. So this is a paper done by Martin at Penn State University a few years ago, where he went out and looked at tropical trees and found out that the R alleles are sort of analogous to MHC in animals. They're important for resistance to pathogens in plants. So if we have a relative abundance and we have some kind of a bottleneck, there's going to be a loss of R gene diversity. And the evidence is that low R gene diversity facilitates spread of pathogens among conspecifics that are susceptible to the same pathogens. So it's really analogous to MHC in animals. And this is a funny kind of comparison. This is not one species, but he went out and looked at six different trees in Barrow, Colorado Island, and looked at the number of alleles at the D genes and found out that species which had a larger NE in this particular area had much more variation at the D locus. So we would think that those species that had larger populations would be more resistant to pathogens than species that had smaller NEs. And the final one that I'm going to talk about is the S locus. So S locus is responsible for self-incompatibility in plants. So plants have a variety of mechanisms so that they don't self. And the S locus, sort of the simplest S locus, the rule is that, so here we have an ovule with the genotype S1, S2. The simple rule is that pollen grains that have the S1 or S2 allele cannot pollinate, cannot effectively produce an offspring. So if we have these 3 pollen grains landing there, it's only the S3 allele which is going to be effective. And so only a pollen grain with a different S allele can facilitate a plant with a particular genotype. So you can see that this is really strong selection for new mutations, because new mutations, the individual that it pollinates cannot have that allele because it's a new mutation. So there's really strong selection for allelic diversity at S alleles. And again, like MHC, even fairly small plant populations often have 40 or 50 alleles because of the strong selection. And this is a paper done by Andrew Young a number of years ago, where they're looking at the button wrinklewort, plants have such great names. And so this is population size of different populations, and this is a number of S alleles. So as we've been talking about, small NE, you're going to have fewer alleles, and as the population size goes from 10 up to 1 million, the number of S alleles increases. And again, the question is, so what? And so what he looked at was mate availability as a function of the number of S alleles. So the fewer S alleles in a population, the fewer mates that can successfully pollinate a seed. So as the number of S alleles goes up, mate availability also goes up. And this has an effect on fitness or seed set. So the more mates that are available, the more effective is seed set. So again, this is a case where having many alleles is important for adaptation. And the other example I used was species in which NC census size is much greater than NE. And this is a review by Sean Hoban and everybody else that worked on conservation genetics published. So they looked at a number of different taxonomic groups, and pretty much they're all the same. The mean ratio between NE and NC was somewhere about 0.3, except for marine fish, which had a NE to NC ratio of 0.024, which means that marine fish, the NC is much larger than NE. And this has been shown, well, not shown, but to support this idea, this is a paper published by Pinsky and Steve Palumbi 10 years ago, where they looked at populations of the same species that were overfished and not overfished. And they found that overfished populations, on the average within the same species, had 2% lower heterozygosity, which doesn't sound like much, but that's equivalent to, remember, you lose 1/2n. So that's equivalent to an NE of 25. 1/50 is 2%. So there's evidence of bottleneck in overfished populations and heterozygosity, but allelic richness was much greater. They found a 12% reduction in allelic richness. And one example of a case where this could have an effect, so herring in the Baltic Sea in 1974 is estimated to have 31 billion fish, and now it's estimated to be 9 billion fish. So one would say, well, 9 billion, that's huge. What's the concern? Well, heterozygosity, there's really no concern. Going from 31 billion to 9 billion, it's less than a 1% loss in heterozygosity. But using that equilibrium that we started with, there would be the number of alleles at equilibrium would go from 5,000 to 1,700. So there's virtually no expected loss in heterozygosity, but a 70% loss in allelic variation. So again, this difference between heterozygosity and allelic variation can be large in some cases. So now I get to the point where I say to myself, so what? I mean, how important is this observation? It's fairly clear that theoretically, allelic diversity is much more common or much more affected by bottlenecks and reduction in population size, which is not taken into account in the standard recommendation of at least NE of at least 500. And I guess the simplest recommendation is that if we're going to use these rules of thumb, NE of 500, we should ignore the fact that this may not tell us everything you need to know about the genetic variation. So looking at the expected loss of allelic variation should be included in these guidelines, as well as looking at effective population size. And this is really only going to be important, well, for loci that have a lot of alleles, like MHC, R, or the S alleles. And it's only good, and also it's going to be important for species that have very large effective populations or very large census sizes relative to their effective size. And so when is this going to be important is either species where NC is really large compared to NE, species that are loci which have a lot of alleles, or species that go through a bottleneck. So if we're dealing with a species that went through a bottleneck, using NE may not do a very good job of telling us what the genetic variation in that species is and how it's going to affect the long-term survival of the species. And just an example, sometimes if we're going to take an action to conserve a species, there may be a conflict between effective size and a number of alleles. So let's think about we're starting a conservation population of fish. And we have often, if we go out and collect wild fish, it's much easier to find fertile, ripe males than females. So should we use all of the males that are available, or once we get up to a few more males and we have females, does it really make a difference? And if we used effective size as the only criterion, we may say using more males is not going to matter because effective size is limited by the number of females. However, if we think about allelic diversity, those males, if we bring them into the population, they might not have much effect on heterozygosity, but they could really increase the number of alleles present in the population. So in the long run, if we're trying to preserve a species by starting a conservation population, having as many males as possible is probably a good recommendation. And as Paulo said, he and I have done a lot of hiking in Montana, and this was a few years ago. I have a picture of Paulo making a snow angel, but Diane suggested it may not be appropriate to put up there. So I'd be happy to answer any questions. Thanks.